and mechanical senses. And the chemical and mechanical basically refers to like the way that the receptors work in these systems. The chemical systems, though, are basically taste and smell. There's a few other sensory systems that are closely related to those, but technically different. And then the mechanical senses are all of the touch-based things. So that would be things like touch, pain, itch. I mentioned last time, like prosopagnosia, your sort of sense of muscles and where they are relative to your body. So those are the kinds of sensory systems that I mean when I say chemical and mechanical. Probably, I'm guessing today we'll maybe only get through the olfactory system, which is your sense of smell. So we're going to start with those. The olfactory and gustatory systems, those are just fancy names for smell and taste. So olfaction is smell, gustation or gustatory is taste. These are probably some of the least well understood sensory systems of all of the ones that we know about. We still have a lot more questions than answers when it comes to these systems as far as like how they work, which is maybe not surprising if you think about it. Like we figured out how to mimic vision and hearing really well. We can make a screen where you can watch videos of like anything you could possibly imagine. I can watch a video of, you know, people across the country, and the visual experience of that is basically the same as reality, right? Same with auditory, right? We can mimic hearing. We have speakers that can create sound. But so far, we can't really make something that can like mimic a smell or a taste, right? If you put on a VR headset, you get the visual experience and the auditory experience, but you're not going to get smell and taste. And that's partially because we just don't really understand how these work very well. Both systems, though, so olfaction and gustation have receptors that are activated by direct contact with specific chemicals. So that's the other interesting thing about these systems, like vision and hearing. You're able to sense things that are really far away from you, right? You can technically see, well, I'll just ask you, how far do you think you can see? Like, yeah, so if you think about it, you can see into space, right? You can see stars that are like light years away. So I can sense something with my vision that is so ridiculously far from me, right? I can hear things that are really far away. Oftentimes, if there's like an earthquake in another city, you know, I might be able to hear that, or like a volcano eruption or something really loud, right? I could probably hear that even if it's like miles away. But I can only smell or taste something if I have direct physical contact with it. You will never taste the sun or smell the sun, probably, right? But you can see it. I guess we can't hear the sun. If the sun exploded, we would probably hear it. But yeah, so you got to have direct contact. So that's the chemical system, and then we definitely won't have time to get to it today, but eventually we'll talk about the mechanical senses, which is also sometimes called the somatosensory system. So that involves all that stuff I was talking about, you know, sensing pressure, pain, itch, proprioception, etc. All right, so we're starting with the olfactory system, which again, olfaction, just a fancy word for smell. So this is a diagram of some of the structures involved in your olfactory system. So obviously you have the nose, which leads into this nasal cavity. And so anytime you smell something, that means you're basically taking particles of it up into your nose, and it goes into the nasal cavity. It's kind of gross to think about, but if you've ever smelled something like nasty, you know, your dog poops and you smell that, the only reason you're able to smell it is because a particle of that is in your nose. It's kind of gross to think about. Yeah. So again, you got to have physical contact. So those particles get up into the nasal cavity, and they're going to interact with these structures called the olfactory receptors. So olfactory receptors, just like photoreceptors or the hair cells in your ears, these are specialized neurons that stick down into the nasal cavity, and basically you're going to pick up any of those chemicals that get into there. And then the olfactory receptors are embedded in this layer of tissue here called the olfactory epithelium. Or sorry, that's not the yellow part, but the part kind of in between. This kind of spongy structure. Really, the olfactory epithelium is just kind of supporting the olfactory receptors. And then if you remember, so do you remember the structure we talked about? It's the first structure in the brain where all the sensory information goes. Anybody remember what that was called? That's the central nervous system is your brain, but there's a structure in the brain. We talked about it last time. So it's where visual information goes first and auditory information goes first. It's like a little structure in the middle. I called it the post office of the brain. I heard someone say it. Yeah, correct, the thalamus. So almost all sensory systems go to the thalamus. The only exception to that is smell. So smell is the only sensory system that does not go to the thalamus. So instead of going to the thalamus, it goes directly to this structure called the olfactory bulb. And then from there, it's going to go to some other structures in the brain. So smell is the exception of all the senses. For whatever reason, it does not get routed through the thalamus. Okay, let's write down some of these structures. So let's start with our title. So this is the start of our talk on chemical and mechanical senses. And I'm putting the date only because I'm in a separate sheet of paper. Okay, so, and first we are talking about the olfactory system. So the structures we've talked about so far are the olfactory receptors. So the olfactory receptors are specialized neurons that, what's the word I want to use? That protrude, let's say. So specialized neurons that protrude into the nasal cavity and are activated by, I don't think I use this word, but I'm going to use the word odorants. So olfactory receptors are specialized neurons that protrude into the nasal cavity and are activated by odorants. So odorants are basically just any chemical that can be taken up by these olfactory receptors. Yeah, so deodorant is basically like neutralizing those odorants. Yeah, it's a good connection to make. So we talked about olfactory receptors. We also talked about the olfactory epithelium. Olfactory epithelium. So that's a specialized layer of tissue in which the olfactory receptors are embedded. So olfactory epithelium is a specialized layer of tissue in which the olfactory receptors are embedded. And then what else? Oh, the olfactory bulb is the other. I knew there was 3 of them on there. So an olfactory bulb, that's a structure in the brain that receives information from the olfactory receptors. I'm going to put a little like asterisk note under this because I'm almost certain that I asked you about this on the quiz, and put that smell is the only sense that does not route to the thalamus. So smell is the only sense that does not route to the thalamus. Okay. So again, keep in mind with taste and smell, we have a lot more questions than answers. So there's a lot of stuff that we don't understand about how the system works. So let's talk a little bit about the little bit that we do know, and then we'll talk about some of the questions that are still kind of lingering. So one of the things we know about smell is that there's a huge range of differences in sensitivity to smells, both across like individual humans, but also across different species. One of the ways that we measure smell sensitivity, there's a lot of different ways to do it, but one is this test where you have like little vials of chemicals that, are odorants, basically a little tiny vial of odorants. And there's actually this like crazy contraption. I should have put a picture of it up here. It's kind of hard to like visualize. I don't know if I could even draw it. Maybe I should look it up real quick. I'm trying to think of what it's even called. Probably like an olfactometer or something like that. Olfactometer. So something kind of like this. I know it's very crazy looking. So you've got an odorant in like a vial here, and that odorant is being sent into this complex tube, and then eventually that shoots out air that is going to go into the nose. So just a way of having like a very contained, purified source of smell, essentially. And what people can do is they can modify the amount of odorant that is going through this device so that you get a very precise amount of odorant going to the nose. And so then you can basically put like, okay, if I put 10 micrometers of some odorant, then can this person detect it or smell it? Do they know that there's an odor there? Also, what's the threshold that they can recognize it? So you might know like, okay, I can smell something, but to be able to know like, okay, that's the smell of oranges or limes or, pine trees or whatever, to be able to name the smell might require a little bit more. So that's just one of the ways of measuring smell sensitivity that has led to all these differences that we know across individuals and across different species. So from experiments using those kinds of devices, we have found that, so within humans, first of all, there's pretty pronounced gender differences in smell sensitivity. So in general, cisgendered women tend to have the most sensitive smell compared to other genders. And that's particularly true for cisgender women who are in like the age range where they could become pregnant. So whenever basically between puberty and menopause, so girls between like 13 and 50-ish tend to have more sensitive smell. And their smell is most sensitive when they're ovulating and particularly when they're pregnant. Which makes a lot of sense. So basically, the part of the job of these chemical senses of taste and smell is to detect chemicals so that we know what we're putting into our body, so that we know, we're avoiding poisons or anything that could be dangerous, and also making sure that we're getting like proper nutrition, that we're getting enough, like sugar and sodium and protein and all the things that we need to make our body run. So a woman who's pregnant is going to be much more sensitive to those kinds of smells because not only is she trying to protect herself, but she's trying to protect this, fetus that's very vulnerable, that if you get even a little bit of like food poisoning or something could really damage the development of that growing embryo. So it makes sense that, pregnant women tend to be the most sensitive to smells.
Do you know why they're so sensitive to like meat and stuff? Because I've just seen a pattern with pregnant women just not being able to handle the smell. Like cooking meat or something like that.
Yeah. I don't know about specific smells, really. I mean, I would guess that it relates somehow to, again, like protecting the fetus, but I don't know the direct, like maybe, meats could potentially carry pathogens like E. coli and salmonella and things like that. So maybe that's what makes us more sensitive. I don't know. Yeah.
My question was, is there a reason as to why some women still have a heightened sense of smell even after they have like a baby?
Probably because, for most of human history, you would then be breastfeeding. I recognize not all women do breastfeed, but like, evolutionarily, that would probably be the mechanism is like.
Years afterwards? Because I still have an aversion, like my son will be 4 and I still have an aversion to hot dogs.
A lot of times when we learn an aversion to food or smell, regardless of how that aversion happens, that's going to last for like a lifetime. So if any of you have ever had the experience of like maybe you eat something and then you get sick after, even if you know it wasn't the food that made you sick, like maybe you just happened to get the stomach flu after you ate this particular thing, like once you've thrown up that food, a lot of us will have like a taste aversion that will last for years. I remember one time throwing up randomly after eating Jimmy John's and like could never eat it again. Even the smell of Jimmy John's now is just like super aversive to me. So yeah, those aversions last potentially a lifetime regardless of what caused them to develop it, to begin with. I think it's just, again, I think it's like an evolutionarily programmed thing that your brain learns like, okay, this food or whatever made me sick one time, and there's a million other options of what I could eat, so I might as well just avoid that for the rest of my life so that I don't ever get poisoned again. It's just like a safety defense mechanism kind of thing. Yeah, Joy?
Based on experience, mostly it's like trauma.
Yeah, it is, yeah.
I cannot eat avocado.
Yeah.
I grew up very poor.
Right.
I eat avocado every day.
Yeah.
I got sick of it.
Yeah.
Now that I'm an adult, I don't want to eat it.
Yeah, so that's often another source of aversion is like, especially if you're forced as a kid to eat something that you really don't enjoy, that can be a traumatic experience that will follow you for the rest of your life. So, yeah. All right, so what else? So we know within individual humans, there's this range in smell sensitivity. And then you all probably are, well aware that across species, there's also a huge difference in sensitivity to smells. So rats, for example, are 8 to 50 times more sensitive to odors than humans. So they can detect, 50 times less of a micrometers or whatever of the odorant. Dogs are up to 10,000 times more sensitive to smells, depending on what the specific smell is. Dog smell is like a crazy thing to think about. I have this cool book that I've been reading. I'm trying to remember what the name of it is. Oh my gosh, I'm blanking. But it's all about like sensory systems in other species. Oh my God, I'll Google it at the end of class today because it's going to drive me crazy that I can't remember the name. But it talks about like, the senses that humans have, but that other species have, more pronounced, like smell and stuff. But then also talks about, senses that other species have that we don't even have, like magnetism, things like that. And it goes into a lot of detail about like dogs' smell system. And it's just really fascinating. It's also changed the way that I think about like when I'm walking my dogs and they stop to smell something. I used to like, kind of rush them along and pull them. But that book describes it in kind of a funny way that like when dogs are peeing on something and then smelling other dogs' urine or marks or whatever, that for them it's almost like social media. That it's like, they smell like, okay, this other dog peed here and I can tell that, okay, that dog ate this thing for breakfast and they've maybe been mating with whatever other dog. Like they can pick up all kinds of information about what that other dog has been doing whenever they smell that. And then when they mark on it, it's kind of almost like liking their post, like being like, oh, I was here too. So whenever you're walking your dog, you want to let them take plenty of time to smell what's around them because that's like their way of, it's like their way of socializing and interacting. So, So, because that's like their most dominant sense, basically. We also know, so back to individual differences in humans, we know also that smell sensitivity changes with age. So typically the older we get, the less sensitive our smell becomes. Which can be sometimes dangerous. So some elderly people might have issues where if they have like a gas leak in their house, they don't smell it because their smell sensitivity is so decreased from what it normally was when they were younger or whatever. So that's another issue.
I have a question.
Yeah.
If an elderly person has dementia, does that make it worse?
I have actually heard that there's a strong relationship that if you start to develop a decreased smell sensitivity, that's like a predictor for developing dementia or Alzheimer's. So, yeah.
The whole house smelled like burnt plastic, and she couldn't smell anything.
Yeah, I've heard that there's a very strong connection between those. I also read, this is kind of also a sidebar and a little bit gross, but I recently read a study that people who pick their nose are more likely to develop Alzheimer's or dementia because you're basically breaking down that olfactory epithelium and sort of like cutting down this barrier between, the world and your olfactory bulb. So it's kind of like a very, vulnerable way that pathogens and things can get into your brain. And yeah, that can apparently lead to like Alzheimer's and dementia earlier. So smell has all kinds of crazy connections. So whenever someone has completely lost the sense of smell, that's called anosmia. So anosmia is a deficit where you have no more smell. Yeah, so this was very prominent during COVID. Some of you might still have anosmia. So that was, one of the long-term effects. Most people who got COVID would lose their sense of smell for maybe like a few days. But then some people would have it that continued and persisted and they never got their sense of smell back. And it's kind of interesting because, I mean, if you ever get like the flu or cold or something, you can kind of lose your sense of smell, but that's more like the nasal cavity is so blocked up that nothing can get in. Whereas with COVID, it was different where it's actually attacking those olfactory receptors. So it's breaking down the receptors. It's not just that it's like clogging up your nose. So that's why some people didn't recover. There's also, I think, like certain medications and other things that can cause anosmia. And I think even like if you have a stroke in the area that's close to the olfactory bulb, then that can also cause anosmia as well. This is kind of different from this idea of individual differences, but we also know there's a very strong connection between smell and memory. So how many of you have ever had that experience where you just like randomly catch a whiff of some smell and it just like you have a vivid memory of something that you haven't thought about in decades or years or whatever? So that's extremely common. There's actually studies they've done to try to test that. So there was a study there where they took people, like older adults, and had them color with like Crayola crayons. And they found that for the majority of people that induced like a very vivid memory of something from their childhood. And part of that might be, so like I said, the olfactory receptors don't go to the thalamus. Instead, they go straight to the olfactory bulb. And then the olfactory bulb is really strongly connected to some structures in the brain. So one of them is the hippocampus, which is a structure we'll talk about a lot more later that has to do with memory. So hippocampus is important for storing memories. And then it also connects to a structure called the amygdala, which is important for processing emotions. Okay, let's stop and write some of this stuff down. So here we're talking about individual and species differences. So women are more sensitive to smells than men. Particularly when ovulating or pregnant. So women are more sensitive to smells than men, particularly when ovulating or pregnant. Let's see, we also talked about how smell sensitivity decreases as we age. We also talked about anosmia, which is the complete loss of smell. All right, then we talked about some species differences. So I told you rats are 50 times more sensitive to smell. And dogs are up to 10,000 times more sensitive. So far we've got women are more sensitive to smells than men, particularly when ovulating or pregnant. Smell sensitivity decreases as we age. We have anosmia as the complete loss of smell. And then for species differences, rats are 50 times more sensitive to smell, and dogs are up to 100 or up to 10,000 times more sensitive. And then the final thing I'll put here, and we'll probably stop there for today. Let's see. Smell is strongly tied to memory. So smell is strongly tied to memory because the olfactory bulb connects directly to the hippocampus, which is spelled like hippocampus, like the animal and then like a college campus. So it connects directly to the hippocampus. I'm going to put in parentheses memory because that's what the hippocampus is responsible for. So the olfactory bulb connects directly to the hippocampus, parentheses memory, and to the amygdala. And then in parentheses, I'm going to put emotion. So smell is strongly tied to memory because the olfactory bulb connects directly to the hippocampus, parentheses memory, and to the amygdala, parentheses emotion. I wanted to talk about in the context of olfaction. So last time we talked a little bit about just kind of, what the olfactory system is. We talked about the structures in the nose, the nasal passages, the olfactory epithelium, all of that. We also talked a little bit about like differences in sensitivity between individual people and also different species. But I told you at the beginning last time that when it comes to olfaction, which is smell, and gustation, which is taste, there are actually a lot more questions at this point than answers. So these are two of the sensory systems that we understand the least about. And so I wanted to talk a little bit about some of those unanswered questions, the things that we still really don't know. So one of the biggest questions that we still don't really understand is how many different kinds of receptors there are for your olfactory system. So if you think about when we talked about vision, for example, I told you there's basically 4 kinds of photoreceptors, right? There's one kind of rod and then 3 different kinds of cones, and that's what lets us see color and contrast, and we understand that really well. But for olfaction, we really have no idea how many types of receptors there are. Some evidence suggests that for humans, there could be potentially hundreds of types of different olfactory receptors. Basically, the way these receptors work is that, these different chemical odorants get up into your nasal passages, and those chemicals will latch onto a receptor. But for them to latch on, they have to match up in really specific particular ways, right? And so the question is how many different types of receptors are there that are responding to those chemicals? And so like I said, different studies have shown that for humans, it could be anywhere in the hundreds or maybe the thousands of different types of receptors. Part of what makes that so tricky to answer is that the way that we sense smells is not really predictable. So if you look at the chemical structure of one thing and the chemical structure of something else, you could have 2 things that have totally different chemical structures, but they smell the same to us. And vice versa, you can also have 2 chemicals that look really, really similar and they smell totally different. And so we just haven't really figured out how the structure of the chemical maps on to our actual perception of smell. So that's still a huge, huge question that we have. And kind of along with that, we don't really know what the property of the molecule is that determines the smell, right? So I don't know if any of you have taken chemistry. It's been a really long time since I've taken chemistry, but if you have taken it, you might remember learning about different structures. There's acids and alcohols and, all these different properties or whatever. We don't really know necessarily which of those properties determines the quality of a smell. And then another huge mystery with olfactory receptors, which to understand why this is a mystery, you've got to understand something about the nervous system. So our brain and most of our neurons are really unique in that they don't typically regenerate. So if you think about all the other cells in your body, think about your skin, for example. It's kind of gross to think about, but your skin cells die really frequently and they kind of float off into the air and create dust and things like that. And then those skin cells are regenerated, right? They reproduce, make new cells that are going to replace those. I think I've heard that every 7 years, you basically have a new, completely new layer of skin because of how often it regenerates, right? Your brain cells are, like the ones that you have now, are the ones that you had when you were developing as a womb in the fetus. Like, like those neurons have not died and then been reproduced or regenerated. You've had them for your entire life. Okay, kind of crazy to think about. So most of our neurons do not regenerate, but olfactory receptors are the exception to this. So if you think about it, like your nose is fairly exposed to the world, right? You're constantly getting like all kinds of irritants. You get a cold or something, anything gets in there and it's going to damage those olfactory receptors. And so because of that, the olfactory receptors actually die often and then are regenerated. But we don't really understand how they do that. Because if you think of part of the reason that you keep the same neurons is think of how complicated it would be. So later in this lecture, we're going to talk about like your somatosensory system. You have a neuron, for example, that goes, one neuron goes from the tip of your toe all the way up to your spinal cord. So think of how long that is for a single cell, right? A microscopic organism to travel that distance is massive, right? So if that neuron from the tip of your toe to your spine, if it died somehow and it tried to regenerate and reproduce, it would have to not only like reproduce, but then it would have to like reform that axon all the way from that exact point at the tip of your toe all the way to the exact same point on your spine. And if anything goes wrong in that process where it's trying to climb up, right, then like your sensation of that toe is not going to be the same anymore. So that's part of why our neurons don't regenerate, is because it would just be too complicated to like rewire the structure the same way. But somehow your olfactory receptors do that. Somehow whenever your olfactory receptors die, they're able to not just reproduce, but then also to rewire themselves up to the olfactory bulb in a way that we still continue to like smell things, you know, relatively the same way. So those are all still unanswered questions we don't really know. Let's write down a couple of the important points from what I just said.
What should I title this part? Is it just...
So we're continuing with what we had last time, so I'm still on the chemical and mechanical, so I would say just start under wherever you were last time.
Okay.
I'll probably put the date off to the side just so I know. You don't have to put the date, though. Uh-oh. Is my Sharpie going to die now? I'm going to try using pen. I don't know if it will show up as well on projector. Okay, so yeah, we're still on olfaction, but I am going to call this unanswered questions. And some of this we're copying a little bit from the slide, but so one of our questions is how many olfactory receptors and really the best answer that we have for that is for humans have, you know, hundreds to thousands of olfactory receptors. So how many olfactory receptors? Humans have hundreds to thousands. Something else I didn't mention, you don't have to write this down, but we know that part of what makes, like I talked about how other species are way more sensitive to smell, so dogs and rats in particular have very sensitive smell, and part of it is that they likely have way more receptors than we do, more different kinds of receptors and also just more overall. So they're like more densely packed within their nose, and then they also have lots of different kinds, so they're able to distinguish more smells than we are. So, you know, I think I read something that rats and dogs probably have like up to like 10,000 different kinds of receptors. And here, just to make it clear, so when I say hundreds of thousands, I mean like the different types, kind of like we talked about with cones and rods, right? But then of each of those types, you would also have lots of them. So, okay. We also don't know, so I'm not even going to put an answer to this one, so we don't know what properties of a molecule determine its smell. And then the last question we don't really know is how do olfactory receptors regenerate? There's probably lots of other questions in addition to those, but those are just some of the big ones. And I should also mention, too, it would be really cool if we could understand this third one, if we could figure out how it is that olfactory receptors regenerate. That could help us to understand how to treat someone who's had like damaged nerves in somewhere else in the body. So if you think about it, if someone ever has like especially damage to the spinal cord, right? If you damage your spinal cord, you're almost certainly going to be like paralyzed, from that damage all the way down. So like if I damage the lower part of my spinal cord, then probably my legs are going to be paralyzed. And like I said, it's almost impossible for your nervous system to then regrow those neurons. If we understood how the olfactory receptors do it, then that might help us to come up with like medications or treatments that could then target other types of neurons to help those regenerate whenever they're damaged. So it's a pretty important question to hopefully eventually be able to answer. All right, so let's move on. So we're technically done talking about smell, but there are some other senses that are highly related to smell, but technically different. So they're different because they use different receptors, and they also take different pathways in the brain. So one of the things you might think of as being very closely related to smell is our sense of pheromones. So pheromones are basically a class of chemicals that are released by an organism, and then they affect an organism of the same species. So, if we think of, when I think of pheromones, I typically think of ants. So if you ever watch like ants in a trail, one of them goes and finds food somewhere, and then, maybe a day later, you'll see like a whole trail of ants going to that same source of food. And the reason that they follow this line, even if it's like really like wonky and curvy and doesn't just go straight to the food source, they do that because the ant that originally found the food will lay down this track of pheromones that basically tells the other ants like, hey, there's food here, go in this direction, right? So it's a pheromone because it's released by the ant, and then it tells the other ants how to behave in some way. So that's essentially what pheromones do. I often see, on Instagram or Amazon or other, like social media and stuff, I'll sometimes see ads for like perfume or something that has pheromones in it. Do you guys ever see that? And the idea is that it's supposed to like target the opposite gender and like attract them to you. So that is complete BS. So it turns out humans basically do not have a perception of pheromones anymore. In our very, very distant evolutionary history, whenever we were like living up in the trees or whatever, we might have sensed pheromones then. But since then, we've completely lost the ability to do that. So pheromones in most species, the information from the pheromones is sent to a part of the brain called the vasonasal organ. I'll write that down in a second, so don't worry about spelling that right now. So the vasonasal organ is what receives this information. In humans, that VNO is like... oh my gosh, I'm blanking on the word. Do you guys know the word for like when you still have an organ but it doesn't function anymore? Like humans still have like a little bit of a tailbone. Oh, no. I feel like it starts with a V, and that's what's throwing me off because I'm thinking vasonasal organ. It doesn't matter. But it's like, it's like we still technically have a vasonasal organ, but it's like shriveled up and tiny and basically dead. It like doesn't respond to anything anymore. It's not. It's like a really weird word. It's not, I don't know. I'll think of it at some point, but yeah. It's like comes from biology. It's like a word that biologists use to describe an organ that you still have, but that's like useless. I don't know. I'll think of it later. So the VNO in humans just like is basically dead and non-responsive, okay? And we also don't have any of the receptors for pheromones. So we know that the receptors that receive pheromones is slightly different than the ones that receive odorants. And humans no longer have those. So we're basically not affected by pheromones. So really anything that you ever hear about pheromones is probably BS. Another kind of similar thing I've heard is that cis women, when they're like together, their menstrual cycle will sync up, and that's from pheromones. Turns out that entire thing is actually BS, that women don't sync up to begin with, and even if they did, it would have nothing to do with pheromones. So not a real thing we have anymore. But other species do have pheromones, and for other species, it's like a very important part of their communication. Yeah.
You said we can't smell pheromones anymore. So at what point, could we, and what happened?
Yeah. I don't know the specific details. I just know that it would have been like before we were Homo sapien, right? So it would have been like when we were like Neanderthal or something previous to that. And I don't really know what would have, I know that humans over time, like our sense of smell has also become less dominant. Probably, I mean, this is totally speculation, but probably what happens is as our eyes and vision became more dominant, we needed more like area of the brain for that. And so there's like a trade-off, right? So it's like, okay, we don't use smell very much anymore, so we can kind of get rid of some of the neurons and structures for that and devote more of that energy to vision. That would be my guess, but. All right, another closely related sense to smell is what's called trigeminal chemoreception. This is also sometimes called the common chemical sense. And basically what this is, it's receptors that are devoted to detecting irritating compounds. So you do have some receptors for trigeminal chemoreception in your nose, so that's why it's kind of related to smell. But you also have these same types of receptors in like your cornea and in parts of your mouth. And this is the sense that is triggered, for example, whenever you're cutting onions and it makes your eyes start to tear up. So basically because you have these trigeminal chemoreceptors in your cornea, whenever you cut the onion, it releases all these irritating compounds, ammonia and all kinds of stuff. And those compounds get into your cornea and it triggers those receptors and basically causes your eye to tear up because it's trying to like flush out those irritating compounds. But again, you also have those in your nose. So if you ever smell something and it sort of gives you like that irritating sensation in your nose, then that's coming from these trigeminal chemoreceptors. All right, so let's write this down. So now, all right, so we're talking about related senses. So first we talked about pheromones. So pheromones are a class of molecules released by an organism and detected by organisms of the same species. So again, that's pheromones, a class of molecules released by an organism and detected by organisms of the same species. And then I'm going to put, just kind of as a bullet point under that, no longer functional in humans due to,
Evolution.
yeah, but I was going to say something about the due to a, let's say, dysfunctional vasoronasal organ. I'm going to put in parentheses V and O because that's often what it's referred to. So no longer functional in humans due to a dysfunctional vasoronasal organ. Vasoronasal is VASERONASAL.
Is that the word you're looking for?
No. It's no.
Okay.
It was something else that started with a V. I think that's why I can't think of it because my brain can only think of that when it thinks of a V word, but I'll think of it when I'm not trying to think of it is what will happen. All right. So then another related sense under that, we have trigeminal, which is TRIGEMINAL, trigeminal chemoreception. So trigeminal chemoreception is a sense devoted to detecting irritating compounds. Trigeminal chemoreception is a sense devoted to detecting irritating compounds. All right, any questions about smell before we move on to taste? Okay, so moving on. So again, taste, the fancy word for it is gustation. So we're talking about the gustatory system. I would say gustation, I think, technically refers to like everything that surrounds eating. So it's not just taste necessarily, but taste is a huge part of it. So again, just like with smell, taste is another sense that we have, probably more questions than answers. I feel like we're making more progress with taste than smell for whatever reason. But yeah, so with taste, obviously the sensory organ that's devoted mostly to taste is the tongue. I will say you actually have taste buds spread throughout your mouth, like the roof of your mouth and your, the inside of your cheeks. I think even the back of your throat has taste buds. But we get the sensation that most of our taste comes from the tongue because that's where they're most densely, the taste buds are most densely sort of collected. So there's a structure or a picture of the tongue here that shows some of these structures. So the tongue contains these little structures called papillae. And papillae are like the little bumps basically on the tongue. So that's what that image is, kind of zoomed in. So if you ever, look at your tongue in the mirror, you see all those bumps on it, those are papillae. And the papillae then contain the actual taste buds. And there's all different types of papillae that have different shapes and structures, and they're kind of distributed along the tongue differently. So you can see those here. So there's the filiform. Filiform are covering the entire surface, and they're these kind of cone shaped, sort of pointy bumps. The fungiform are more mushroom shaped, and they're on the sides and the tip of the tongue. The foliate are folds at the back and sides, and then the circumvallate are flat mounts that are at the very, very back. So you really only see those if you stick your tongue way out and you kind of look at the very back. But all those are just different shapes, like I said, of papillae. Each papillae then contains a bunch of taste buds, and each taste bud contains a bunch of taste receptors. So we're kind of zooming in to the bumps, then you see the taste buds, and then you zoom into those taste buds, and that's where you'll see the actual taste receptors. And I hope you guys all realize by this point that anytime you use the word receptor, that's basically referring to a specialized type of neuron. So the olfactory receptors were a type of neuron, the photoreceptors were a type of neuron, taste receptors are also a type of specialized neuron that basically stick up out of the tongue, or they're sort of embedded in these taste buds actually, and they basically sense chemicals. So the way that the taste receptors work is actually very similar to the way that the olfactory receptors work. So you have, chemicals that get into the mouth and they bind to these taste receptors, and only very specific chemicals will unlock a specific type of receptor and make it trigger and fire, and then give you the sense of taste. I'm trying to think of what we should write down from here. I don't think I asked you very many quiz questions about this stuff, but maybe. Okay, so we're starting a whole new topic here. All right, so here we're talking about gustation. And right now we're talking about structures in the gustatory system. I think really all we need to write down from here is that the tongue contains papillae. It's P-A-P-I-L-L-A-E. Tongue contains papillae, which contain taste buds with many taste receptors. So the tongue contains papillae, which contain taste buds with many taste receptors. All right, I would also say just like with smell, we don't really know for sure exactly how many taste receptors are, although there is a lot more agreement about how many with taste compared to smell. So remember with smell, I told you there's potentially hundreds to thousands of receptors. With taste, we have thought for, historically, for most of the history that we've been studying this, most people thought that we had 4 different types of taste receptors. And this is, again, one of those things you might have learned in like elementary school or something. I remember when I was in elementary school doing some sort of like little experiment where we like dipped, cotton swabs in like different flavors, like lemon juice and sugar water, and like rubbed it on our tongue. Did any of you do that? I don't know if that was a Midwest thing or a millennial thing or like what, or maybe just my school randomly did it. I don't know. But yeah, so you might have learned that you have these 4 different taste receptors: sweet, sour, bitter, and salty. And there is wide agreement among people who research gustation that yes, we definitely have at least these 4. But then beyond that, there's a lot of controversy about are there more than 4, and like what exactly do they respond to? So a likely 5th candidate that pretty much everyone agrees on at this point is a 5th taste called umami. So I think we probably all are very familiar with the first 4, right? Everybody knows what sweet, sour, bitter, and salty taste like. Umami you might not be as familiar with if you're not into like cooking, or especially if you haven't explored like Asian cuisine. Umami refers to this flavor that's kind of like savory. So if you imagine the taste of like mushrooms or meat, things like that, then that would be sort of what umami is. And like I said, there's quite a bit of agreement that we probably have at least that 5th. I should say, so each of these receptors responds pretty predictably to different classes of compounds. So sweet, obviously, those receptors respond to sugars, so things like glucose, fructose, sucrose, etc. And then we also now have this whole class of like non-nutritive sweeteners, things like aspartame, that your sweet receptors respond to. Sour responds mostly to acids. Bitter responds mostly to alkaline substances that could potentially be poisonous. And then salty responds mostly to sodiums. So those are fairly predictable in terms of like what they respond to. There's also quite a bit of evidence that we evolved these 4 flavors for very specific reasons. Or I should say tastes. I should be careful about the words I'm using. So these are tastes, not flavors. So these 4 tastes, it's theorized that they evolved to help us to get the nutrients that we need, and then also to avoid things that could potentially be poisonous or harmful. So sweet is very important. We need sugar in our diet because that's what gives us energy to be able to do all of the things that we do. It's what powers most of our cells. Sour, so it turns out we also need a lot of acids to help with, different chemical processes in the body. Bitter mostly probably evolved actually to avoid poisons. So we have things in our diet that are bitter that are not harmful if you think of like cocoa powder or coffee. But in our evolutionary past, most of the things we would have encountered that were bitter were probably poisonous or bad for us. And then salt is also, sodium is really important. It's, necessary for like powering our neurons and things like that. So the idea is that we evolve the ability to detect these chemicals because they have really important properties. So part of the controversy, I think, of umami is that it's less easy to come up with an evolutionary explanation of why we would need this 5th receptor to detect umami. I think all of us would probably agree that protein is important in our diet, but umami doesn't exactly correspond to protein. That's what we often associate it with, is things like meat, but it's not just meat that will trigger that. In fact, the thing that most triggers the umami receptors is MSG, or monosodium glutamate, which is a type of sodium. So you would think that that would activate our salty receptors, which I think it does to some extent, but it mostly activates these umami or savory receptors. So then beyond those 5, there's a lot of controversy about, do we have more taste receptors besides those? So these are some possible candidates for maybe 6th, 7th receptors, but there's not a lot of agreement if they actually are separate receptors. So things like metallic, some people think of like onions, for example, have kind of sometimes like an irony or metallic taste. Creamy would be mostly like dairy products. Astringent is, I think of it very closely related to sour, but it's slightly different. So if you think of like cranberries, for example, they have that kind of like astringent quality. And then pungent is also possible. I don't remember like what the food correlate for pungent is. Maybe cheeses, things like that, things that have that really like intense flavor. But yeah, so those are the likely receptors for taste. So let's write these down. So now, how many tastes? All right, so we know we have sweet receptors. So sweet receptors detect sugars such as glucose, fructose, and sucrose. So sweet receptors detect sugars such as glucose, fructose, and sucrose.
One's just like white sugar.
I think sucrose. I'm not 100% sure. And it might even be a combination. So most of the sweet things that you eat would have a combination of those. All right, sour receptors detect acids. Sucrose, S-U-C-R-O-S-E. All right, so sour receptors detect acids. Bitter receptors respond to alkaline substances. That may be poisonous. Bitter receptors respond to alkaline substances that may be poisonous. And then salty receptors detect sodium. All right, we should also put umami is a possible fifth taste that responds to MSG. Another kind of myth that I learned in elementary school that turns out not to be true is that these taste receptors are like distributed in different spots on the tongue. Did you all ever learn that? Like I remember learning, I don't remember what it was, but like sweet is on the tip and salty's on the sides or something. It turns out that actually these receptors are spread pretty much evenly across the tongue. So I don't know where that myth came from, but it's not true. Another thing I'll say, have you guys ever heard of super tasters? So super tasters are people that have like a really sensitive taste sense, basically. And they also, there's like a very specific molecule that they're able to detect that other people can't. And all I remember is that it has something to do with like the density of the receptors. Basically, people that have a more sensitive taste palate just means that they have more of those taste receptors sort of clustered more densely on the tongue. And that's what makes them more sensitive. All right, there are lots of chemicals that can alter the way that these taste receptors respond. Some of these you might be familiar with, you might have experienced it. So one of them actually is monosodium glutamate, or MSG. So I told you MSG is the molecule that activates umami most strongly. So it's used in a lot of like Asian cuisines. But it also, so MSG basically, let me try to remember the logic of it. It essentially makes your salty and sweet receptors more sensitive. And so what that means, if you put a little bit of MSG in something that has salty and sweet flavors, then it will like enhance those tastes and make them taste even more salty and sweet than they would without the MSG. And again, that's because they're actually making it so those taste receptors respond more vigorously to like a smaller amount of substance. Another molecule that can alter your taste is sodium lauryl sulfate, or SLS. This is a chemical that's often used in toothpastes. It's an ingredient there. So how many of you have ever had the experience of like brushing your teeth and then you, I don't know, take a drink of orange juice or something like that and it just tastes absolutely horrible? So the reason that the orange juice tastes really horrible is because of this compound, sodium lauryl sulfate. So basically what SLS does is it inhibits the sweet receptors. So the sugar in the orange juice is basically like your tongue isn't detecting that at all, so you don't taste the sweet of the orange juice. And then it also makes the bitter receptors more sensitive. So it's enhancing all the bitter taste of the orange juice, getting rid of the sweet, so that's why it makes it taste so horrible. And then the third one you should know about is a substance called miraculin. So miraculin is a compound that's found in these little berries called miracle fruit. Any of you ever heard of miracle fruit? So basically this miraculin does kind of the opposite of sodium lauryl sulfate. So what miraculin does is it makes the sweet receptors more sensitive, but it also alters the structure of your sour receptors. No, sorry, hold on. I got that backwards a little bit. It inhibits your sour receptors, and then it alters the sweet receptors so that they will respond to sugars, but they will also respond to acids. So instead of your sour receptors responding to acids, your sweet receptors respond to acids. And basically what that does is it tricks your brain into thinking that sour things taste sweet. So if you eat a miracle fruit and then you can just drink like a shot of lemon juice or straight vinegar and it'll taste sweet instead of sour. It's kind of a crazy experience. So you can actually so in Iowa, whenever I taught there, I taught like a higher level class that was all sensation and perception. And I had like a whole day where I brought in miracle fruit tablets. So you can buy them on Amazon. They're really cheap. They're just like a little tablet and you kind of suck on it so it coats your tongue. And then I brought in a bunch of like sour foods for people to taste. Because it's kind of a crazy experience. Because like I said, you can, take like a warhead or some candy that's just like crazy sour. And it doesn't taste sour at all. It tastes more sweet than it normally would. So that's miracle fruit. I've heard of also like restaurants that have this as kind of like a gimmick that they can give you miracle fruit before they bring out the food or whatever, and all their food is like tailored to have some acidic things in it that activate this miraculin or whatever. Okay, so let's write those down. So these are chemicals that alter taste. So monosodium glutamate, or MSG. So MSG increases sensitivity to sweet and salty. So MSG increases sensitivity to sweet and salty. What was the next one? Sodium lauryl sulfate. Which is SLS. So sodium lauryl sulfate inhibits sweet and increases sensitivity to bitter. Sodium lauryl sulfate inhibits sweet and increases sensitivity to bitter. And then finally, miraculin. So miraculin inhibits sour and makes sweet receptors respond to acids. So we've got Miraculin. Aculin. It's possible like Word or something might not have that saved as a word in the dictionary. All right. So regardless of how many taste receptors we have, whether it's 4, 5, 6, whatever, you might recognize that if we only had those tastes, then probably we wouldn't be able to experience the like range of flavors that we do, right? If you think of like an orange versus a lime versus a lemon, all of those are basically sour, right? And maybe I guess an orange would also be maybe a little bit sweet. So if we only had taste, then we probably couldn't tell the difference between a lime and a lemon and an orange and a grapefruit and all these different, and then also probably couldn't tell that from like vinegar or, something else. So flavor is this broader experience that we have of food that's a result of many senses coming together. So flavor is a result of mostly taste and smell. So if you think of any time you've had like a cold or the flu or something and your nose is stuffed up, then you lose a lot of your sense of taste, right, is what it seems like. Like you eat something and it just doesn't have the same taste it does. Really what's happening when you have a cold or flu is mostly that you're losing your sense of smell. Your taste buds and taste receptors and everything are still intact, but if you take out smell, then food loses a lot of its flavor. So, if you think about it again, like the way that taste and smell work are very similar. Their receptors are both responding to chemicals that are binding to those receptors. And they're also even like physically close in space. And in fact, like our nasal passages basically connect our mouth and our nose to each other. So whenever you eat something, you're smelling it first because, to bring it up to your mouth, it's going to be close to your nose. But then also, like all the odorants are getting into your nose through that like passage that goes sort of in the back, almost like the back of your throat, up to your nasal passages. And so that's what gives us this experience of flavor. Flavor can also be affected, though, by things like temperature and texture. So it turns out that our taste and smell receptors are most effective whenever we're eating something that's roughly at like room temperature. So if something is really hot or really cold, then our taste and smell receptors are going to have a harder time sensing the compounds in that substance. So, if you've ever been like eating ice cream and you get down to the very end of it and there's like the melted part at the very end, a lot of times that will taste sweeter. Not because it's actually sweeter, there's not any more sugar in it, it's just that it's gotten to room temperature, so your sweet receptors can kind of like sense the sugar in it a lot better than it would whenever it's really cold. So temperature can affect flavor, texture can also affect flavor. I've heard of lots of people, for example, who really hate tomatoes, but they like pizza sauce or ketchup or something that's basically made of tomatoes. And the reasoning I've heard is that most people, whenever they bite just into like a raw tomato, the texture reminds them of like a rotten fruit, which it basically, right? Like that texture is very similar to if you have like a peach or something that's way overripe. And so because the texture is weird, it tricks their brain into thinking that it tastes bad when it's not really the taste, it's really the texture that's affecting the sort of experience of flavor.
I've been that way my whole life up until the last year. I've started eating small tomatoes.
Oh yeah, the little cherry tomatoes. Yeah, I've always loved tomatoes, but I totally get like the concept of the texture. I feel like cherry tomatoes are better because they're like so small and they just like sort of pop.
I think it was just the acidic taste of them I didn't like.
Yeah,
Like
yeah.
the texture, I can't do like the hard shell and the licking bush.
Right. Yeah. Yes. Yeah. I get it. I love them, but I get where that is coming from. So the idea though is that the flavor of a food is really the result of all these different senses coming together. And if you actually look at structures in the brain, there are areas in the brain that will only respond if they're getting input from like at least two of these systems. So there are some areas of the brain that are just for taste, some that are just for smell, But then there are other areas that have to be getting input from both of those at the same time in order to be activated. So from the tongue, information is sent to the thalamus. So remember, every sensory system except for smell sends their information first to the thalamus. Then it goes to a structure called the gustatory cortex. So that's for taste. But then taste and smell combine in all of these different areas. So in the olfactory bulb, that's getting information from the smell receptors, but also from the taste receptors. And then some structures called the insula in the orbitofrontal cortex. The orbitofrontal cortex is like the front part of the brain that sits just right above the eyes. And then also the amygdala, we talked a little bit about last time. The amygdala is really important for like emotional processing. So if you think of all the emotional connections that we have with food and smell, right? There's probably recipes that have been passed down in your family, or maybe something that your parents cooked for you when you were a really little kid. And like if you eat that meal, you get really nostalgic and you have all these emotional memories, right? That's because the amygdala is a structure that's connected to both taste and smell. And then also the hypothalamus. So the hypothalamus is a structure that regulates a lot of our like metabolism. So it's sort of what tells us when we're hungry or when we're full. And so obviously like taste and smell, right? You can feel full, but then you smell pizza or brownies or something, and that smell basically will tell your hypothalamus like, yeah, I know your stomach's full, but actually you really want this food. And so it might make you feel hungry even though your stomach is actually full, because you're getting activation from the hypothalamus. Okay, all right, let's write some of that down. So here we're looking at taste versus flavor. I think for this one, basically just copying the slide. So flavor is the result of taste, smell, temperature, and texture. So flavor is a result of taste, smell, temperature, and texture. And then from the tongue, gustatory information is sent to the thalamus. And then the gustatory cortex. So from the tongue, gustatory information is sent to the thalamus and then the gustatory cortex. And then next, we'll write down structures that respond to taste and smell. So structures that respond to taste and smell, we have the olfactory bulb, my pen does not want to stay like I wanted to. Okay, olfactory bulb, insula, I'm just going to put OFC, but that means orbitofrontal cortex. Amygdala, and hypothalamus. So there's olfactory bulb, insula, OFC, amygdala, and hypothalamus. All right, any questions about taste? All right, we're at about that. Okay, so next up, we are going to switch to talking about what's called the somatosensory system. So this is one of the mechanical senses. And mechanical refers to the facts. Remember, chemical senses are taste and smell, and they're called that because it takes a chemical binding to the receptor in order for them to activate. Mechanical senses are activated mechanically. So things like pressure and vibration and stretching are what will activate these somatosensory receptors. So there are lots of different kinds of somatosensory receptors, and they are embedded in these different layers of the skin. So, skin is actually your largest organ. If you think about it, it covers your entire body. So it's like this massive organ. And it's composed of 3 different layers. So the epidermis is the outer layer that you can see. Epidermis is mostly like dead skin cells at the very top layer. And then living skin cells, are embedded right underneath that. Then under the epidermis, you have the dermis and the hypodermis, and that is where the somatosensory receptors are found. So they're found in those 2 bottom layers. The dermis is this kind of soft layer that basically just connects the epidermis and the hypodermis. And then the hypodermis is where the majority of your fat cells are stored. So the hypodermis is going to connect the rest of the skin to your muscles and your bones, and it stores energy in the form of fat. So there are, like I said, lots of different mechanoreceptors. We're going to talk about, probably a handful, maybe 6 to 10 of them today. And they all have very different physiology, which is what allows them to respond to this wide range of different types of sort of mechanical stimulation. So some of the things that determine what a mechanoreceptor will respond to. So first of all, they have different nerve endings. So these are, again, specialized neurons, and they have different, so some of them just have a bare nerve ending. It basically just doesn't have anything on it. It's like a soma with no dendrites or anything. Some of them have these really elaborate structures. You'll see pictures of some of them on the next couple of slides. So that nerve ending changes the way that they respond. They also have different thicknesses of their axon. So remember, the axon is the tail-like structure that sends the signal. The wider the axon is, the faster it will respond. And then the thinner it is, the slower it will be. Another thing that affects the speed of the neuron is whether it's myelinated or not. So if you remember, I think last week we talked about the structure of neurons, and I told you about myelin. Myelin is only on some neurons, but not others. And this is where you will see one of those differences. So neurons that are myelinated are faster than those that are not. What else? Another thing that's affected by the physiology of these is what's called receptive field size. So receptive field size is the area of skin that a neuron responds to. So if you think of your fingertips are pretty sensitive. So if you're ever trying to do really fine, movements like writing or typing, you're using mostly your fingers, right? So they're very sensitive compared to your forearm or your back or the back of your leg. Those areas are less sensitive. And part of the difference in sensitivity is how densely packed those receptors are and the field size. So on your fingertip, each little receptor is responding to just a little teeny tiny area. Whereas in those other areas of your body, your forearm or your back or whatever, each neuron is getting information from a giant patch of skin. So that changes their sensitivity and what they can respond to. So yeah, and then also their depth in the skin. So you can see, I don't know if it really shows any, you can kind of see some, here's some mechanoreceptors in here, and then there's some down here. So how close it is to the surface of the skin will also determine what it's going to respond to. So on the next few slides, we're going to go through a few of these different mechanoreceptors, and I'll point out some of those physiological differences, and we'll talk about what exactly are they responding to in the environment. So first, let's write down some of these structures I just listed off in the skin. So now we are talking about the somatosensory system. All right, so the somatosensory system uses mechanoreceptors embedded in the skin. So it uses mechanoreceptors embedded in the skin. Let's list off the important structures of the skin. So one of the layers that's important is the dermis. So the dermis is below the epidermis. And contains mechanoreceptors. So the dermis is below the epidermis and contains mechanoreceptors that respond to pressure, stretching, and vibration. So the dermis is below the epidermis and contains mechanoreceptors that respond to pressure, stretching, and vibration. And then the hypodermis is the bottom layer of skin. That contains fat cells and mechanoreceptors. So those two layers of skin, the dermis and the hypodermis, are the important ones that we're going to focus on. And then we should also put, so mechanoreceptors. Mechanoreceptors physiology varies in axon diameter, myelination. So I'm basically just listing off all the features that can vary. So mechanoreceptors physiology varies in axon diameter, myelination, receptive field size. What else did I talk about? Depth in skin. And I'm just going to use the word encapsulation. So that's encapsulation, encapsulation. That's referring to what I was talking about, like the end of the neuron, whether it's just like an open free nerve ending or if it has like specialized structures on it. So mechanoreceptors physiology varies in axon diameter, myelination, receptive field size, depth in skin, and encapsulation. Yeah, Marcus. Myelination is whether it's got that like fatty tissue wrapped around the axon. If it does, it acts kind of like, oh my gosh, what's the word? I keep blanking on words today. Kind of like a wire that's covered in rubber. Yeah. So basically, if it has myelin, it's going to go faster compared to if it does not have myelin. Does that make sense? So the myelin kind of helps like hold the electrical charge into the axon so that it can go straight down instead of like leaking out everywhere. Maybe. I don't know. I'm trying to think of the word for when you have rubber around it. Right. Insulation. That's the word I was trying to think of. I don't know why my brain is struggling today. Yeah, it's like insulation on a wire. This is how you should think of it. The myelin. Okay. Yeah. I assume you're an electrician or something. You do something with electricity, obviously. Yeah. Yes.
Doing that since '88.
Yeah. You would probably understand a lot about neurophysiology then. It was always weird taking neuroscience classes because like half of it was basically physics. Like,
Just got done doing lifeguard cert this weekend.
yeah.
So yeah, it's funny how all this crosses over.
Yeah. Okay, so we've got, like I said, these two different layers of skin. The dermis and the hypodermis is where these mechanoreceptors are going to be found. So now we're going to talk about some of these different receptors. And these all have just like the wackiest names ever, some of them. I assume that all of them come from like the person who discovered them, but some of them are kind of silly sounding, especially the next slide. These ones aren't so crazy, but the next ones are. So two of the types of mechanoreceptors are called Merkel receptors and Meissner corpuscles. Corpuscles is a fun word that you'll hear quite a bit. And these are found in that higher layer of skin in the dermis. So remember, epidermis is the very outer layer. We're not going to see any mechanoreceptors there, but the next layer under that is the dermis, and that is where you find these Merkel receptors and Meissner corpuscles. And these basically respond to different types of pressure. So the Merkel discs in particular are really responsible for detecting like really fine detailed texture. So, you know, if you're like feeling the difference between something furry and soft versus like a rough brick surface or whatever, the difference between those you're detecting mostly with these Merkel discs. The Meissner corpuscles, again, very fun word, are mostly responsible for detecting like your grip and pressure sort of like across the skin. So think of the Merkel receptors as more like fine detail on your fingertips, whereas Meissner is like things that are going across the skin or gripping. So whenever I like grip my, water bottle, the ability to sense that difference like in pressure across my skin to know that I'm gripping it is coming mostly from the Meissner corpuscles. So some of the properties that they have, so I already told you they're high up in the skin. So they're really close to the outer surface. And they both have these pretty elaborate structures on the end of their, well, the Meissner corpuscles are pretty elaborate. The Merkel discs are not so much. So the Merkel receptors are the red here. So they're sometimes called Merkel discs because the top of them is just sort of like this disc shape. It's almost like a cone with like a disc, almost like a suction thing or whatever. Is kind of what they look like. And then you can see the Meissner corpuscle is the blue here. So the red is basically like the dendrite of the neuron. And then all the blue stuff surrounding it, that's where it gets the name corpuscle. It's like all these little kind of fluidy structures that are surrounding the neurons and kind of cushioning it so it changes the way that it responds. So they're close to the surface of the skin. They've got some interesting encapsulation going on. You don't need to worry about this stuff about firing. You can ignore that. That's part of the picture that I put up there. So. They also have very small receptive fields. So they're packed really tightly and densely in like your fingertips. And I mean, they're found all over your skin, but I associate them with the fingertips mostly just because that's where I think of like fine detailed stuff. But they're really small receptive fields, so they're packed really densely and really tightly. In general, women tend to have more sensitive like fingertips compared to men. And part of it is that women tend to have more of these, the Merkel receptors and the Meissner corpuscles in our fingertips, sort of like packed really tightly. That increases sensitivity. So let's write these down. So for pressure perception, okay. For pressure perception, we have the Merkel receptors. So Merkel receptors are found in the dermis, close to the surface of the skin. Responds to fine, detailed textures. So it's found in the dermis, close to the surface of the skin, It responds to fine detailed textures. So that's the Merkel receptors. Then the Meissner. So Meissner has two S's. Meissner corpuscles. These are also found in the dermis. Found in the dermis, encapsulated.
What's that word? The first one, messy.
Meissner, Meissner.
Meissner.
Yep, Meissner corpuscles, found in the dermis, encapsulated by, I'm trying to think of how to describe these. Encapsulated by jelly-like blobs, very scientific, jelly-like blobs. And respond to grip. They respond to grip and motion across the skin. Okay, so Meissner corpuscles found in the dermis, encapsulated by jelly-like blobs, respond to grip and motion across the skin. All right, so next up we're going a little bit deeper into the skin. So these next structures, again, have very fun names. So we have Ruffini cylinders and Pacinian corpuscles. So Ruffini cylinders have this cylindrical shape. So you can see those here. So again, you've got kind of like the neurons, and then it's got this kind of blue cylinder. It's not blue in real life, but in the picture, it's got this cylinder kind of almost like fiber structures that run perpendicular to the dendrites of the neuron. So that's the Ruffini cylinder. And then the Pacinian corpuscle also got, so corpuscle I think of as like layers of encapsulation. So you can see the Pacinian corpuscle, it's got, again, that kind of jelly-like, but it's more in like an onion structure where it's in layers instead of kind of just the blobs that we saw with the Meissner corpuscle. Okay, so let's see. The Ruffini cylinder mostly senses stretching of the skin. So if you think of if you like try to stretch your fingers as wide as you can, and you can kind of feel that stretching of the skin, that's your Ruffini cylinders responding to that. So you can think like as you do that, you're basically pulling these little fibers are going to pull really tight. And those little dendrites sticking up into those fibers are going to sense that. So that's the Ruffini cylinders. The Pacinian corpuscle is responsible for detecting more like vibrations. So if you think of as you're like stepping, you can kind of maybe feel the vibrations in your skin, or if you like hit something, like you're going to feel the hit, but you also feel like the vibrations sort of radiating through your skin. So that would be the Pacini corpuscles that's sensing that. And so again, these are found a little bit deeper. So they're kind of in like so the Ruffini cylinders are in the dermis, but they're deeper down. So you can see like the Merkel discs are up here. The Ruffini corpuscles are up like right, almost touching the epidermis basically. Whereas the Ruffini cylinder are in sort of the middle of the dermis, and then the Pacinian corpuscle is way down in the hypodermis. So these are much deeper in the skin. They also have much larger receptive fields, so they're not going to be as sensitive to like detailed fine texture. It's more like large scale vibrations and pressure that they're sensing. All right, so this is stretch and vibration. So the Ruffini cylinders, does it have two Fs? Yes, Ruffini with two Fs, Ruffini cylinders. So these are found deeper in the dermis. So Ruffini cylinders found deeper in the dermis, comma, encapsulated by long fibers. Detects stretching of the skin. So stretch and vibration perception, Ruffini cylinders found deeper in the dermis, encapsulated by long fibers, detect stretching of the skin. And then next we had the Pacinian corpuscles. So Pacinian corpuscles are found in the hypodermis. Encapsulated by onion-like layers. And respond to vibration. So Pacinian corpuscles found in the hypodermis, encapsulated by onion-like layers, respond to vibration. We should also, if you have room up where you had Merkel and Meissner, maybe under that put that both have small receptive fields. So this is for Merkel receptors and Meissner corpuscles. I forgot to put that. So both have small receptive fields. And then under the Ruffini cylinders and Pacinian corpuscles, put that both have large receptive fields. Pacinian. PACINIAN. So Merkel receptors and Meissner corpuscles both have small receptive fields. Ruffini cylinders and Pacinian corpuscles both have large receptive fields. Okay, so we've got these different mechanoreceptors embedded in the skin. And think about it, so these are spread all throughout your skin, covering your entire body. And then, all the axons of those bundle up into lots of different nerves. And those nerves eventually travel from the skin to your spinal cord. And, it's in a pretty organized fashion. So it's kind of like I was saying, like all the things from your lower body are going to eventually connect up to the lower part of your spine. And then as you go up, the higher parts are going to connect to the higher part of your spine. So because of that, like I was saying earlier, if you ever damage your spine, especially if you completely sever your spine, then you're going to be paralyzed from there all the way down. So if you were to, paralyze yourself or to break your spine up at the very top, then you're basically going to be quadriplegic. You're going to be paralyzed all the way from the neck down. And that's because you're severing this connection of the spinal cord to those different areas. So from the spine, eventually, again, that information is going to go to the thalamus and then eventually make its way to the somatosensory cortex, which is in the parietal lobe. So that's what receives that information. Another thing to note is that all of this is crisscrossing, so everything from the right side of the body goes to the left side eventually. It kind of varies where it crosses, and I can never remember off the top of my head. So we'll talk in a second about pain receptors, which are like a totally different type of receptor, and they have different nerves that enter the spine. And one of them, it's either pain or this stuff, the somatosensory stuff we were just talking about, one of them crosses over in the spine, and then one of them crosses over in the brain, and I can't remember which is which. So, but either way, by the time it gets to the brain and you're sensing it, it's the right side of the brain controlling and sensing the left side of the body, and vice versa. Let's see, what else do I want to say about these pathways? I think I already said everything I wanted to earlier, so, because we talked a little bit about the spinal cord and like the fact that you have this giant, axon that travels all the way from your toe to your spine. Pretty interesting stuff. So let's just write down that important stuff real quick. So the somatosensory pathway. Do I have it in my notes which one crosses first? No, I don't. All right, so let's see. Nerves travel from the skin to the spinal cord. So nerves travel from the skin to the spinal cord. From the spine, information goes to, let's see, I'm going to say from the spine, information goes to the, I'm going to use this word contralateral. Contralateral is the fancy way of saying what I was saying earlier, where it crisscrosses. So contralateral means that the right side is going to the left side is going to the right. So from the spine, information goes to the contralateral thalamus, and then to the somatosensory cortex in the parietal lobe. So from the spine, information goes to the contralateral thalamus, and then the somatosensory cortex in the parietal lobe. All right, I actually do have it in my notes. I was trying to remember which one crosses because it results in this kind of interesting pattern of deficits if you have damage to your spine. So the touch information stays on the same side through your spine, and then it crosses once it gets to the brain, whereas the pain information crosses immediately. So what that means, there's very rare cases where someone has just damaged, like let's say they damaged the right side of their spine. So if they damage the right side of their spine, that means they're going to lose touch in the right side of the body, but they're going to lose pain in the left side of the body. So you get this really weird pattern of results for someone who has damage to the spine in this really specific way, where it's like half of their body is feeling one thing but not the other, and then the other half is vice versa. And it's, again, has to do with when that information crosses to the other side. All right, I'm trying to decide. I want to make sure we leave time for the quiz. So I think we'll stop there. So next time we'll talk about pain and itch, which are two totally different receptor systems compared to what we've talked about so far. Yeah. Okay. Okay, that's fine. Thanks for letting me know. Next thing we needed to discuss then are some different receptors that are found within the somatosensory system. So far what we've talked about is mostly what probably most of you think of as touch. So like pressure and, vibration and things that you feel on your skin. But also under this umbrella of the somatosensory system are some other types of receptors that are responsible for sensing things like pain, itch, and also temperature. So let's see. So with pain, there's actually 3 different types of pain that have completely different mechanisms that cause them. So one of them is inflammatory pain. So that's caused by whenever you have damage to your tissues, then that inflammatory response will cause pain, swelling and things like that. There's also neuropathic pain. So neuropathic pain is caused whenever you have damage to your nerves. So, if you have like brain damage that causes a stroke or something, that can sometimes result in pain. If you have damage to like your neurons in a particular muscle, like carpal tunnel syndrome, for example, people who experience pain from carpal tunnel syndrome, it's because the actual neurons have been damaged, and so it causes pain basically. But the type of pain that we're mostly going to focus on today is what's called nociceptive pain. So nociceptive pain is caused by firing of these neurons called nociceptors. And nociceptors are, they're just basically specialized neurons that respond to painful types of stimuli. So there are two broad types of nociceptors that are called slow and fast pain receptors. And the slow and the fast, the name of those refer both to the speed of the actual neuron firing, but also kind of like the sensation of the pain itself. So if you think about, like, let's say you hit your thumb with a hammer. So you smash your thumb and it has that sort of like throbbing pain. That type of pain is mostly encoded by the slow pain receptors. And if you think about that pain, it's kind of like long lasting. It lingers for a long time. It has this sort of slow sensation to it. Yeah, like that throbbing sensation, yeah. Whereas fast pain receptors, those would respond more to like if you stabbed yourself or poked yourself with a needle or something like that, something very sharp that, like you feel it immediately. And typically that type of pain will also go away fairly quickly compared to something like, hitting your thumb. One of the physiological differences that causes that difference between the slow and the fast receptors is just whether or not it's myelinated. So remember, myelin, it's like the insulation on the cord of your neurons. Fast pain receptors have myelin, and they also have slightly thicker axons, so that allows them to send that electric signal faster, whereas the slow pain receptors do not have myelin, and they're also a little bit thinner, so they tend to actually send like a slower signal. And then in addition to pain receptors, we also have itch receptors, which are physiologically very different, and they also take a totally different pathway and then eventually end up in a different area of the brain. It does turn out, though, that there is a relationship between the itch receptors and the pain receptors. So if you think about it, so if you have something itchy on your skin, what do you usually do? You scratch it, right? Which is kind of weird if you think about it. Like why would we scratch at something if it's itchy? And there's actually two main reasons that that's kind of our, instinctive response to something that's itchy. One is that a lot of times itchy is caused by like a chemical compound, some sort of irritant that's on the skin. So if you think of like a mosquito bite, right? Part of the reason that that's itchy is because whenever the mosquito bites you, it releases a chemical that has like irritating compounds. And so when you scratch, that could potentially remove that compound. It's like you're scraping it off to get rid of it. But also, one of the bigger reasons that we tend to scratch when something is itchy is that pain receptors actually inhibit our itch receptors. So if you think about it, scratching is almost like painful, right? You're like scratching yourself, you're inflicting pain on your skin, and that pain inhibits the itch receptors so that you feel the itch, sort of less compared to when you're not scratching it. It's kind of similar with rubbing and pain. So if you think about if you like bang your knee on the corner of something, oftentimes our reaction is to like rub it, right? And that's because rubbing activates the pressure sensors we talked about last time, and those pressure sensors inhibit the pain receptors. So there's like this interesting relationship between all these different mechanoreceptors where the different kinds kind of inhibit each other. All right, then we also have temperature receptors. I'm not going to say much more about that. So they're receptors that respond to like hot and cold substances or different temperatures. And again, those are totally separate receptors. They have completely different physiology, and they also take a separate pathway eventually to the brain. So most people who study this stuff, they tend to think of these all as separate senses. So instead of just having touch, we actually have pressure, and then we also have pain, itch, and temperature as all different sensory systems. Okay. So right now we're talking about pain, itch, and thermal perception. All right, so we have 3 different kinds of pain. Three different kinds of pain. There's inflammatory. Does inflammatory have 2 M's? I think it does. Yeah. Inflammatory. Neuropathic. And then nociceptive. Nociceptive has all C's and no S's, so it's NOCICEPTIVE.
NOCICEPTIVE.
Yep, nociceptive. And then under nociceptive, you should put that there is slow pain, colon. So those slow pain is caused by thin, unmyelinated nociceptors. So nociceptor is NOCICEPTORS. And so nociceptor is just a specialized neuron that's specifically coding pain. So slow pain is thin, unmyelinated nociceptors. And then fast pain is thicker, myelinated nociceptors. And then I think really the only thing we need to say about itch and temperature is just that they're separate from pain. So I would put itch and temperature receptors. So itch and temperature receptors have different physiology. And take different pathways to the brain. Okay, so far we got pain, itch, and thermal perception, 3 different kinds of pain, inflammatory, neuropathic, nociceptive. And then under nociceptive, we have slow pain, which is thin, unmyelinated nociceptors, fast pain, which is thicker, myelinated nociceptors. And then itch and temperature receptors have different physiology and take different pathways to the brain.
I didn't get the fast pain ones.
The fast pain is thicker, myelinated nociceptors. Also, just to make it clear, when I say different pathways to the brain, I basically just mean they're sort of bundled up in a different cable of nerve, but they're still going to go to the spinal cord and then eventually to the thalamus and then to a specific area of the somatosensory cortex. So in the somatosensory cortex, there's all these different senses kind of going there, but routed to different areas of that part of the brain. So. All right, next let's talk a little bit about pain relief. So obviously we have all kinds of different medications and techniques to try to relieve pain. Some of these are maybe more common than others. And really what pretty much all of these do is they're targeting the neurotransmitters that are released by the nociceptors. So the nociceptors are the ones we just talked about that code for slow pain or fast pain. And the main neurotransmitter that those neurons release is something called substance P. It's kind of a weird name for a neurotransmitter. All the other neurotransmitters we've talked about, things like dopamine, serotonin, I don't know, but this one is called substance P, is what's released by nociceptors. And so a lot of these different chemical compounds, they basically target that neurotransmitter and inhibit it or block it in some way. Or they might have other effects that are basically just inhibiting those nociceptors in some way. So things like NSAIDs, non-steroidal, anti-inflammatory drugs, those are targeting inflammatory pain mostly, which again, that's like your inflammation response I talked about earlier, but they also do have some impact on those nociceptors to like reduce their functioning a little bit. Opioids and endorphins, they actually block substance P more in the brain. So instead of targeting the nociceptors themselves, they're sort of working more like upstream and inhibiting that substance P once it's getting to the brain. There's also some evidence that cannabinoids can potentially be effective in relieving pain, particularly more like in the peripheral nervous system. So they basically block, they might also have effects on the inflammatory response. There's a lot less known about how cannabinoids work compared to some of these others. But one of the main things that it probably does is inhibit that inflammatory response. This is maybe kind of surprising. So any of you know what capsaicin is? I don't think we've talked about this yet, which is kind of surprising. Yeah, so capsaicin is what's found in hot, spicy peppers. And it's what gives spicy peppers their spiciness, basically. So capsaicin, I'm surprised I didn't bring it up whenever we talked about taste, because actually capsaicin, we often think of spicy as a taste. But really what capsaicin actually does is it activates our thermal receptors. So the reason that we think of spicy food as hot is because it's literally activating the hot receptors in our tongue and in our mouth. So it's not really activating the taste buds, it's activating those thermal receptors, which are completely separate. And there is this relationship. So before I talked about how, like if you itch, that causes pain, and the pain inhibits itch. Well, we also know that the thermal receptors can inhibit pain. And so if you're activating the thermal receptors, then that can, reduce your pain response. So there are some compounds that contain things like capsaicin, or they might go the opposite route and contain something like menthol. So menthol has sort of the opposite effect. Instead of triggering your heat receptors, it triggers your cold receptors. So if you think of like Icy Hot, for example. So Icy Hot is like a cream that you can buy if you have sore muscles. You can rub it on there and it'll help like relieve the soreness. I think there's actually even a specific like type of Icy Hot. It's like not the normal one, but there's like a specialized one that actually has capsaicin in it.
doTERRA like oils or whatever.
Yeah.
They have one called Deep Blue, and I use that. It's really good.
Oh, really? Yeah.
You can't get it on your like eyes to wash your hands with soap, so you gotta like burn
Right.
the crap out of you. So.
Yeah. Yeah. So, but all of those, they have some sort of compound that's activating the thermal receptors, which again then is going to inhibit your pain response. All right. A really interesting kind of effect is what's called the placebo effect. You guys ever heard of the placebo effect? You probably have some idea of what it is. So placebo effect is basically where you take some kind of substance that's inert. Usually placebo is going to be something like a sugar pill. So it has literally no chemical compound in it that should interfere with your brain in any sort of way. It's just like taking a sugar cube, basically. And yet, if you give that to someone and tell them that it has pain-relieving properties, then a lot of times people will report that their pain symptoms will go away or at least be reduced. So that's called the placebo effect. Most of the time, I think people tend to think of the placebo effect as like, it's like a fake thing. It's like, oh, you take this placebo and you say you feel better, but somehow, it's not real, and so you must be faking it somehow or something like that. But it turns out actually that whenever you give someone a placebo and you tell them that it has pain-relieving effects, researchers have found that the way that your brain responds to that placebo is actually the same as the way that it would respond to taking something like opioids or endorphins or any of these other pain-relieving compounds. So something about the placebo actually tricks your brain into thinking that it should work, and so then your brain responds as if it is working, and then it does what it's supposed to do, basically. So placebos, even though there's no actual chemical compound in them causing anything, they actually do have this effect that's very similar to other types of pain relief. All right. think that's all I wanted to say about that. So let's switch over here. All right, so pain relief. Let's see, I'm going to say many substances that relieve pain. Many substances that relieve pain work by inhibiting nociceptors or substance P. So many substances that relieve pain work by inhibiting nociceptors or substance P, comma, which is a neurotransmitter released by nociceptors. All right, so many substances that relieve pain work by inhibiting nociceptors or substance P, which is a neurotransmitter released by nociceptors. And then under that, I'm just going to list, so that would include opioids. Just so you know, too, I guess I didn't say it. So opioids, would include like heroin, but it would also include things that we use medically. So things like morphine, for example. You know, you commonly will get like morphine or other types of morphine-related pain relievers if you are in a medical setting. You know, you get a surgery or something like that. So that's what I'm talking about with opioids. So opioids, endorphins. Endorphins, I don't think I said this either. Endorphins are like your body's natural. They're very chemically similar to opioids. That's actually where the name endorphins come from. It means endogenous morphines. But they're released naturally by the body, but they're chemically very similar to opioids. So opioids, endorphins, and cannabinoids. And I'm just listing those under there because those are the ones that work the way that I just said. So those inhibit substance P. Okay, so then others work by activating thermal receptors. Which inhibit the nociceptors. So others work by activating thermal receptors, which inhibit nociceptors. And then under that, I'll put capsaicin. And then let's also just list icy hot. So you have an example that's kind of more, that you're probably more familiar with. So pain relief, many substances that relieve pain work by inhibiting nociceptors or substance P, which is a neurotransmitter released by nociceptors. So that would include opioids, endorphins, and cannabinoids. Others work by activating thermal receptors, which inhibit nociceptors, and that would include capsaicin and icy hot. All right, moving on to our final type of sensation that falls under this umbrella of somatosensory system. So far, all of the receptors that we've talked about are mostly embedded in the skin. So remember we talked about like the dermis and the hypodermis and all those pressure receptors, and also a lot of the like pain and itch and thermal receptors are also in the skin. Now we're talking about receptors that are found much deeper, actually like in your muscles or somehow attached to your muscles. And these receptors code for a sense that's called proprioception. So proprioception is kind of my awareness of like where my limbs are relative to the rest of my body. So I think I mentioned this before, but like if you touch your finger to your nose, especially if you're doing it with your eyes closed, the sense that you're using to be able to do that is proprioception. And there are two broad types of receptors that code for the stretch and tension of your muscles and allow you to have this sense of proprioception. So they're called muscle spindles and Golgi tendon organs. All of these have such fun names. So the muscle spindles are pictured in yellow. They're the ones here that kind of spiral around the length of the muscle. So the muscle spindles, basically, if you think about it, whenever you stretch your muscle, it's going to cause that little coil to also stretch, and that will cause it to activate, basically. And then whenever it contracts, that neuron can also feel that as well, and it will inhibit whenever it's contracting, basically. And so through that mechanism, then you know how your muscles are stretching in space. The Golgi tendon organs are also pictured in yellow, but they're the ones that look more kind of like, like little gecko feet or something. So those are attached to single spots on the muscle, and they're more sensing like the overall tension of the muscle. Like if you're, lifting a weight and your muscle is really tense, then it's going to activate those, whereas if your muscle is more relaxed, then the Golgi tendon organ is not going to be activated. So those are the two that mostly code for proprioception. So let's write those down really quickly. So this final sense is proprioception. So proprioception is your awareness of the location of your limbs. It's easiest to think of this as the limbs, but I guess proprioception would also include, like, if you think of just your posture and your balance, like where your trunk is and, things like that. So it's not just your limbs, but that's kind of one of the main things it does. So it's awareness of the location of your limbs. So we talked about muscle spindles. So muscle spindles are proprioceptors that coil around the length of a muscle. So muscle spindles are proprioceptors that coil around the length of a muscle and code for stretching of the muscle. And then Golgi tendon organs. Golgi tendon organs are proprioceptors that attach to a single location on the muscle and code for tension of the muscle. So Golgi tendon organs, proprioceptors that attach to a single location on the muscle and code for tension of the muscle. All right, I'd also point out maybe on this diagram, so the yellow ones, those are the ones we talked about, muscle spindles and Golgi tendon organs. Those are sending information to the brain to tell your brain where your limbs are in space currently. And then, of course, there's also nerves that come from the brain and go to the muscle that basically tell the muscle to move. And so those neurons are often, sort of almost talking in like a cycle so that you're constantly updating the position of your limbs so where they are so you can do whatever you're doing. Like if you're taking a step, that requires that your brain constantly knows where your muscles are, but then is sending signals to tell those muscles how to move. So it's like this very cyclical interaction between those neurons to keep everything like coordinated and synced up, all of that. All right. Last thing I want to talk about for somatosensation is what's called phantom limb syndrome. Any of you ever heard of phantom limb syndrome? It's a pretty bizarre condition. So phantom limb syndrome affects mostly typically people who have had some sort of amputation and have lost a limb. So let's say, for example, someone for whatever reason has had maybe their arm amputated. And after that arm is gone, they sometimes will have the sensation that they can still feel that arm. For most people who have phantom limb, the sensation is typically going to be painful. So they will have just like these phantom pains where it feels like suddenly they're having like a cramp in their muscle or something like that, even though the arm doesn't exist anymore. So that's what phantom limb syndrome is called. There's occasionally people that I've heard of, it's much, much, much more rare, where people can have phantom limb of like a limb that they never had to begin with. So they might feel like they have like a third arm or something like that. And there's also one that's the opposite that's really bizarre, where people will have all their limbs intact, but they will like mentally reject their limb. So there's one case I read about in a book by a neurologist where someone had this condition where like they would be sleeping at night and they would fall out of the bed because they would be trying to push their own leg off because they thought it wasn't their leg but was someone else's. So it's kind of like the opposite, where like their leg is there but they feel like it shouldn't be. It's a really bizarre one. So I want to focus, though, on phantom limb of people who have had amputations and kind of talk about what the theoretical cause of that probably is. So this map here is laying out, it's sort of a map of the somatosensory cortex. So the somatosensory cortex is this strip. It sort of overlaps between the parietal and the frontal, like right on the border of those two lobes. And basically it's laid out to where like very specific parts of the somatosensory cortex respond to certain parts of the body. So if someone were to touch my face, then this area of the somatosensory cortex is what's going to be activated by that sensation of my face being touched. Whereas if I touched my thumb, then it's going to activate this area instead. It's kind of interesting that from all the research that's been done, it seems like most people have the exact same map. So, from person to person, you can pretty easily find like what is the area of the somatosensory cortex that's going to respond to the face, because it's basically the same from person to person. Couple things you might notice about this that's kind of strange. So first of all, the map is not proportionate to the body. So you can see that the face takes up this really huge area of the somatosensory cortex compared to something like your trunk is very small, right? Where like for our body, the face is much smaller than the trunk, but the area of the brain is not proportionate to that. This is part of the reason why your face is so much more sensitive than your trunk. If you think about it, like, your face, very sensitive area. And that's because there's so many neurons in your brain devoted to processing the feeling of your face, basically. You can also see that it's not exactly laid out like the body either. So kind of the body, I guess, is laid out as it normally would be, I guess, like in space. But then you've got this weird thing where like your face, what codes for your face is located directly next to your thumb, which is kind of strange, right? So what happens in someone who has an amputation? Let's say they have their arm amputated, right? So their arm is cut off. Let's say that happens when they're 40 years old. So for 40 years, their brain has been receiving information from this arm, and it's been going to this really specific part of their somatosensory cortex, and that's how they've lived their whole life. Now you cut that arm off. So this part of the brain is not just suddenly going to disappear, right? It's still there. And the brain is often very like hungry for sensation. The brain wants to have input and stimulation. And when it's not getting that from its normal source, it will look for that stimulation from anywhere it can get it. So often what will happen in people who have had an amputation is they have this reorganization of their somatosensory cortex, where the part that used to respond to the amputated limb is going to kind of be taken over by the neighboring areas. So over here you see that the face area has basically stretched to cover that arm that's been amputated. So then what can happen is if I touch the face of the person whose arm has been amputated, then they're going to get activity to this face area, but then that's going to spread to this area that used to code for their arm. And their brain is going to have a hard time figuring out, is that sensation coming from the face or is it coming from my arm? Because for 40 years, if there's been activity here, that means there's something touching my arm, right? And so, yeah, so you get this sort of spillover activation from other areas of the body that causes these sensations in the amputated limb. There's a couple things that can help treat phantom limb. One of the biggest ones is just to basically give the person a prosthetic. So if you give them a prosthetic, and especially then there's like very specific training that they do. There's one, I'm trying to remember the exact setup of it. There's this one study that I saw where they basically took someone who had their arm amputated and they give them a prosthetic. So now they have like a fake plastic arm. And of course that, the prosthetics, especially have been used in the past, they're not going to have any sensation. It's not like I touch that plastic and you're going to feel it. But they do this thing where they have them like, there's like a mirror in between their two arms. And so they're looking at, I don't remember the exact setup, but somehow they're like looking at the arm that's amputated in the prosthetic, and then they're like touching their other arm with something, like rubbing a feather on it or like drawing on it a pen with or whatever, but they're looking at it in a mirror so that it looks like that sensation is happening on the other arm. It's like this weird thing. And it basically doing that sort of like trains the person to feel the prosthetic, even though, they're not getting actual stimulation from any neurons or anything. And so in that way, it kind of like tricks the brain into thinking you still do have an arm, even though you really don't. And that can help sometimes reduce some of the sensations from phantom limb. All right. Any questions about phantom limb? Okay. Let's write down really quickly basically just what phantom limb is. I think is really all you need to know for that. So phantom limb syndrome. So phantom limb syndrome affects, I'm trying to remember the correct version of affects those with an amputated limb and results in sensations from the missing limb that are often painful. So phantom limb syndrome affects those with an amputated limb and results in sensations from the missing limb that are often painful. And then let's write one more little sentence about this. It's likely caused by activity in the somatosensory cortex. So it's likely caused by activity in the somatosensory cortex. Spreading from a neighboring area of the body. So likely caused by activity in the somatosensory cortex, spreading from a neighboring area of the body. Okay, any questions about somatosensation, touch, pain, itch, proprioception, any of it? Okay, I think we'll take a short break here so then we can get into learning about learning.